All right, so I'm Sam Mengi, and my uh, person that I chose for the biology role model project is Brian Kobielka. So as he was growing up, he first became interested in science when he saw because he wanted to be a physician. He really liked his local pediatrician that he would go around his town, his small town he grew up in, and he saw that there's lots of respect for him, and he respected him very much as well. So that's why he first became interested in going into science. He also really liked his math, physics, chemistry, and biology classes in high school, which kind of led to him going into the sciences. He first really became involved in science when he was studying biology at UMD with the intent of going to medical school. During his freshman year, his biology professor allowed him to work in his lab. So he started off by cleaning glassware and doing bench work, but he eventually moved on to become a more major part of the lab and learn new things, which really got him into the chemistry science. And he's also did some summer research projects with an organic chemist at UMD and a research project at a Malaysian lab later on in his career. But these things all helped him really start. These are the first things he really first became involved with science at. Besides, after he graduated from UMD, he did get into Yale Medical School, where he went to become a doctor, because that's what he wanted to do at first. And then after that, he went to residency in St. Louis, Missouri, and then finally he did his fellowship at Duke University, where he first started doing his research. Afterwards, he worked at Stanford, where he found his major findings and stuff. But these were the main places he went to school. And the main obstacles that he had to overcome on his journey to becoming a scientist were in elementary school and grade school. Growing up, he had a very hard time reading, so that was something he had to overcome. Also at Yale, he was very culture shocked because coming from UMD, like a smaller public school, he was not ready for all the Yale and Ivy League schools that Ivy League school kids that are going to be there. He also had a number of financial constraints for medical school and for housing afterwards in Stanford. So he had to take lots of scholarships and do community service to get scholarships. And he also had some trouble with his getting results when he was doing his fellowship at Duke. So those were some of the main obstacles that he had to overcome in his journey towards becoming a good scientist. Some of his interesting hobbies that he has are biking, which he's gone to Yellowstone and back as a kid, and he's also done a lot of running in his time as a scientist, which he'd normally run to and from work when he was back at Duke for his fellowship, which was 5 to 10 miles each way, normally a day. So the thing that I most admire about him and why I chose him is that he won the Nobel Prize in 2002 in chemistry for his studies of G protein coupled receptors. He had many difficulties in his lab work at first, but was able to persevere and get through them, which is something I really identify with and hope to be like someday. I also enjoyed my math and science courses very much more than anything else in high school, and I'm currently enrolled in studying biology as undergraduate and trying to get into med school. And we'll have a research project at the U of M as well as part of my requirements as an honors student. So those are some of the reasons why I really identify with him. What he's currently working on and what his field of biology is that he actually works in is cell communication, or more specifically, G protein coupled receptors on cells. So these are basically the receptors that of adrenaline, and they are very important to how our cells are able to communicate with each other inside our bodies. And he is currently focused on characterizing the structure and mechanism of activation of G protein coupled receptors, which he has done through a variety of approaches, including cell biology, gene disruption in mice, and in vivo physiology using a spectrum of biochemical and biophysical tools. The most recent paper that he is an author on is called Trehalose cord amphiphils for membrane protein stabilization, the importance of detergent micelle size and GPCR stability. So for this paper, the experiments they did where they studied diverse membrane proteins and they tried to find the correct detergent to efficiently extract and stabilize the proteins from the native membranes for analysis. So they had to use a novel class of trehalose cord amphiphils with multiple alkyl chains and carbohydrates projecting from the core to be able to figure out the GPCR stability. And so what they did for this, they prepared two sets of trihaloose colored detergents with either a glucoside or maltoside hydrophilic group directly attached to the trihaloose core. And for an additional set of amphiphils, 8 glucose units were attached to the central trihaloose unit using a glycerol linker. Introduction of the glycerol linker allowed them to prepare the water-soluble TCGLs with alkyl chain length to extend the C-carbon-12. And using glucoside and maltoside head groups along with the glycerol linker, they varied the detergent alkyl chain from C5 to C12. And so from this, they're able to find that micelle size was very important in actually the stability of the GPCR proteins. So the main techniques that they used for this paper included DNA amplification, transmission electron microscopy analysis of the data, and NMR spectra, density function theory calculations, and NTA affinity chromatography, which are all very important to analyze the data and see the different proteins to the human eye. And then, so the main things that he's discovered in his career are successfully identifying the gene which regulates the formation of the receptor for the hormone adrenaline, which is the GPCRs. And he also discovered that the receptor was similar to receptors located in the eye that capture light, and that there is indeed an entire family of receptors that look and act in similar ways, which are the G protein coupled receptors. So that's what he won the Nobel Prize for, which is why he's famous and his most major success. But the next steps that he's still taking today are he's looking into the structural insights of the activation of metabolic GPCRs. He's trying to figure out how to characterize the structure and mechanism of the activation of G protein coupled receptors. And he's also trying to find the contributions of protein dynamics to the functional versatility of GPCRs. So clearly, Brian Kobilka is a very important scientist in history, and his work today has been very helpful because GPCRs actually are very key towards most medications today. Almost half of them use GPCRs to implement their effects on humans. So his work has been very key to society. And then here's a list of my references that I used for this video.